So I think we'll start the session for today. The sharing my screen, so please let me know whether the screen is visible or not, and whether my voice is audible clearly. Anyone just let me know whether everything is working fine or not. Okay. Yeah. So I had preponed the class for this Monday. So we'll cover what Professor Pomiha went over in week 9, and we'll also just take a brief look at what we did or what we talked about in the earlier classes for week 8. So again, I welcome all of you to the week 9's live tutorial session for the NPTEL course Bioinformatics Algorithms and Applications, instructed by Professor Michael Gromiha. So can anybody quickly brief me what Professor Gromiha went over in week 9's lecture? We'll try to cover some concepts regarding those and try to see some questions, I guess.
Sir, I was not able to complete the whole week as I planned to do in this Saturday, Sunday. But however, I watched 2 to 3 videos which were about protein folding rate and protein types of protein interactions, how protein interactions takes place, like such as protein DNA interaction, protein interaction, and how the mutations are changed according to that not mutation due to that interaction their values change and sometimes it can be more or sometimes it can be less, such as ASA or long range order or something like that. Also he talked about protein folding rate, how fast or slow protein can unfold. I think so it's about that.
Yeah, so different concepts regarding protein folding as well as protein interactions, right? Interactions with DNA or protein interactions.
Yes. I was not able to complete the last 2 videos.
Yeah, it's fine. It's fine. So yeah, so we'll talk some concepts regarding those. We'll try to solve some questions. I think you have seen these types of questions in some of the earlier classes as well. But we'll go forward with that. Okay. So in week 8, particularly to briefly talk about what we did. So we are talking about the whole structure of PDB, right? And in the PDB structure, of course, you have the coordinate information. So you have different flexibility information. For example, in B factor, you have the atoms and numbers, and the residue identifiers and whatever atoms, every corresponding information regarding those, as well as your occupancy information, those kind of things are given, right? So the PDB structures, of course, can be utilized in a variety of calculations. For example, if you have distance calculations, if you have multiple conformations, you want to see what is the difference between those conformations, we can calculate RMST. We can see the compactness parameters, we can see the solvent accessibility. So there are different types of calculations possible. And of course, in the terms of the interactions of proteins and ligands, it could be protein DNA, protein nucleic acid, protein, right? So those interactions also, if you want to compute those, you will need the help of PDB structures or certain structures. If it is not X-ray crystallographic structure, you have to build it. You have to model it from ground up, so that you can use this for different tools, right? In different tools for calculating your binding or your interactions, right? So those we'll go over. So briefly, yes, we talked about RMST. RMST is basically the deviation parameter that allows you to see the structural difference or the conformational difference between different, let's say, different states or different conformations. One would be your reference, one would be your target, right? Widely useful in assessing not only the deviation, but also in certain cases, for example, in simulations, to assess the convergence. Convergence means if your system has become stabilized or not. So it's a good parameter to know. We also talked about the moment of inertia and the majorly the associated radius of gyration. So the radius of gyration was your compactness parameter basically. So if your radius of gyration is bigger, so that means that the protein is less compact, right? If your radius of gyration is smaller, then the protein means that the protein is more compact. So again, you can use these sort of parameters to assess the broad scale structural changes as well as the local structural changes. So for example, if your protein is from an unfolded state, it's going to a folded state, you will notice that its radius of gyration is diminishing, right? It's reducing. Similarly, in the case of a particular local environment or a local sort of structure where you might want to let's say investigate certain motives, right, and their confirmation. So in that case also data visualization can give you useful ideas uh, about how your structure is shifting. So it's again a good useful information to know. We talked about stabilizing residues. So stabilizing residues have certain different parameters, for example, surrounding hydrophobicity. So if your hydrophobic residue is buried in a hydrophobic environment, then that will be quite stable, right? However, conversely, if your hydrophobic residue is sort of slightly exposed to a polar environment, or even if your polar residues are buried in a hydrophobic environment, so those kind of uh, local environments will give rise to instability. They will increase the energy of the overall system, and the system will become unstable, right? So for that, certain calculations are there. For example, just the overall stability metrics or parameters. You have surrounding hydrophobicity, you have long range orders, you have certain stabilization centers, and also you have conservation scores, right? So those are certain things that you take into account when you look at the overall stability parameters in a way. And then of course, you saw certain problems which uses free energy terms, right? So free energy terms can be used to describe uh, changes occurring after mutations. For example, what this particular question was asking about in average assignment method. So valine to alanine mutations, multiple are given. If you are asked a different sort of valine to alanine, you can do average or mean calculation of all of this, all of the available data, and then you will get the free energy change in a general sense of what a valine to alanine transformation energy looks like, right? Mutation induced energy changes. So you can look at that. Free energy change is also quite useful. I guess we will talk about some of those in the current lectures also. We looked at the average assignment method, and also we looked at some flexibility parameters, for example, p factors and normalized p factors, where if it is greater than 1, then it's a flexible sort of residue. If it's less than 1, then it's a more rigid residue, right? Flexibility and rigidity, and you can assess it to basically investigate the parameters or the fluctuation parameters of particular residues on a residue level. Here you can basically take multiple atoms into account for a particular residue, and you can assess their fluctuation level, right? Yeah. So we looked at the different characterization and categorization. And finally, we looked at different mutations, right? So mutations, depending on what type of mutations they are, and as well as depending on the characteristics of the mutated residue and the surrounding context, basically surrounding environmental context, they can either stabilize the whole structure or they can destabilize the whole structure. So we looked at different metrics. For example, here, if it is increasing in size and if there is already a tight packing, then the mutation will induce instability or destabilize the structure, right? Similarly, if there was an important salt bridge formation which was mutated, one of the residues are mutated to an opposite charge, so then that will destabilize the structure. If uh, there is a tight packing and a residue is mutated to a bulkier residue, then it will destabilize further. However, if there is already a loose packing and the mutation that causes the bulkiness to occur, it kind of fills up the space. So then the space filling mutations could sometimes be stabilizing set of mutations, right? And again, depending on these different contexts, we can also look into um, the free energy change following mutation, and we can correlate that with what type of surrounding environment the residue was present beforehand, right? So that was one type of analytical question that we looked into earlier. And talked about some sort of interactions, for example, van der Waals calculated by the Lennard-Jones potential. And that was basically most of it, right? Yeah. So in this particular lecture for week 9, as you said, the major focus was on protein interactions, right? Interactions between different proteins, protein ligand interactions, protein DNA nucleic acid interactions, so on and so forth. And then Professor Kumiha also went over the concepts regarding protein folding, right? Fast folding proteins, what are their compositions, slow folding proteins, folding kinetics, and there was a plot used to describe that as well, right? So these different types of concepts Professor Kumiha went over. So let's try to quickly recap. I think I have talked about this earlier also. So what is the following terms? in a protein ligand complex, and here we are specifically talking about the binding, right? The binding of a protein and a ligand. So three terms are given here. One is binding site, another is binding mode, and another is binding free energy. So what could these be? Binding site, binding mode, and then binding free energy. Then write in the chat also. So binding site, what does that mean? Binding site, from the term you can guess, it is the particular position of the protein or the particular pocket of the protein where your ligand is binding, right? So that is the binding site or binding region or binding pocket where basically your interacting amino acid residues are present, which stabilizes your complexation with the ligand, right? So that is called the binding site. Then another is binding mode. What could binding mode be? So is binding site different to binding mode? What are you talking about here? Binding mode. So remember that in protein ligand complex, of course, for example, if you do a molecular docking, oftentimes what you will see is that for a particular set of protein and ligand, you will get different results, right? You will get multiple results for the particular complex that has been formed following docking, and you will have the corresponding scores or the free energy scores or the docking scores for each of the pose that has been created, right? So binding mode essentially indicates exactly that. It is the geometry of the ligand at the binding site. So due to the different binding modes that are possible, we will see that the different results from the docking that will be listed one after another. Okay. And then they will have the corresponding energy value for each pose, for each of the geometry of the ligand at the binding site. There could be different geometries that have similar score. So you have to choose amongst those. But that is exactly what the binding mode is called. It's basically the pose of the ligand at the binding site. Okay. And finally, we have the binding free energy. So does anybody can anybody tell what will generally be the binding free energy in terms of its value? So should it be a positive value or should it be a negative value? In a just in a normal protein ligand complex, let's say you have performed your docking. So what is the likely outcome of the binding free energy? Is it a positive value? Is it a negative value? And what does binding free energy indicate? Negative, yes. So negative value you will get usually for the binding free energy calculations. And what the binding free energy indicates, it's basically uh, the energy change or the change in energy levels kind of uh, in the complex following complex formation. So initially, let's say you have a change in energy after binding and before binding. Yeah, so let's say initially you had one protein, you had one ligand, right? Individually, they have certain energy levels. Then after you have bounds the ligand to the protein. So now the protein ligand complex is formed, right? And this whole complex afterwards that has a certain energy level, right? So if your final minus initial, so after binding and then before binding, that energy difference is negative. That means that the binding reduces the energy level of the entire system, right? So reduces the energy level. That means that certain energy is liberated, let's say, and then the system is becoming more stabilized. So your final minus initial value for that will indicate your binding free energy. And the more negative the binding free energy is, usually it's the better, uh, because that means your complex is more stabilized, sort of in energy terms, right? So it's the free energy of complexation, and can also indicate the binding affinity. So there are also formulas to convert your binding free energy values to binding affinity, different binding affinity terms. For example, let's say your dissociation constant, KD value, right? You can convert it, your binding free energy using the formula del G equals to minus RT ln KD. Okay, so that will give you an energy to sort of KD value comparison or correlation kind of, right? So you can do that, but this is one formula which is called the Gibbs free energy formula, right? So delta G equals delta H minus T delta S. So what is this delta H, this T, and this delta S terms, and what does delta G indicate? So delta G is basically your binding free energy, and these are kind of the constituting, the broad constituting parameters for your binding free energy, right? So what does delta H and delta S indicate here?
Sir, delta H represents the enthalpy change, and the delta S, I guess it's the entropy change, and T is for time, temperature. Sorry.
Yes, temperature. So delta H is the enthalpy change of complexation. Delta S is the entropy change, and then T is the temperature, absolute temperature. So this overall, this minus T delta S, this term is your entropic contribution to the binding free energy, which is also important to calculate to properly assess the complexation, right? So here you see that delta G equals to G complex minus G receptor plus G ligand, what I talked about earlier. So the complexes energy minus the sum total energy of the receptor and ligand individually. If you do this calculation, you will get the binding free energy delta G value, right? And here one let's say representative figure is given, and this is basically a protein, right? As you can see through this ribbon structure, the different loop structure, secondary structures, and so on. So that is particularly a protein, specifically the binding pocket of a protein. So the binding region of a protein. You will see different residues they have their side chains visible, right? Side chains highlighted. Okay. Uh, so these residues, since they are a part of the binding site, kind of, for the visualization, their side chain has been highlighted, the side chain orientation, so on and so forth. And then you will see also the ligand present here, right, in this yellowish color, right? So this ligand present at the binding pocket, it also has a certain conformation, right? So for example, its ring structure is positioned here, and then it's the other chains that is sort of oriented in that way, right? So that is a particular pose or mode of the binding. So this is overall the what we're looking at is the binding site, the binding region of the protein, and then the ligand conformation or the ligand pose that will indicate the binding mode of the protein of the ligand, right? Binding mode of the ligand in this complex, in this particular complex, right? If you have different ligands, of course, docked to the same protein, you will see that uh, they might have different sort of preferred binding poses or binding modes, even if let's say the difference between the different ligands are minimal, right? For example, it's only the change of 1 or 2 atoms from this ligand, right? Even still, it could happen that the binding mode is changed. For example, the atoms and the chains that was oriented this way might not again oriented in the same manner if you change a couple of atoms here and there. Okay, in the base preferred poses. So binding mode can change, binding poses can change. Of course, what you have to assess the strength of the binding via that would be your energy terms, right? The binding free energy. So there are different calculations, of course, apart from docking. We have talked about molecular dynamics simulations, right? So in molecular dynamics simulations also, you get a trajectory, basically a behavior of your system. Let's say it's a protein ligand complex, and then you can calculate the binding free energy. Okay, so I have talked about this before also. So do you remember what was the method, or what was one of the methods that can be used in calculating binding free energy from a molecular dynamics trajectory? So one method that we have talked about uh, that was called MMPPSA, if you remember. Okay, molecular mechanics Poisson Boltzmann surface area. So the MMPPSA calculation, if you do, that gives the enthalpic contribution. You saw the delta H component of the binding free energy. So the delta H component you can calculate by MMPBS, and an associated calculation which is called normal mode analysis, that will give you your entropic term. Okay, so this example I think I have gone over earlier also. So this is the energy component that you get from MMPBS. It constitutes of different subcomponents. For example, your van der Waals interaction, electrostatic interaction, polar solvation energy, non-polar solvation energy, uh, and the different delta G gas, so gas phase energy or solvated phase energy. That will be basically, if you combine these different terms, you will get these results. But your delta total or your total enthalpic term, that comes from this van der Waals electrostatic polar and non-polar solvation free energy taken together. And then if you combine the translational, rotational, and vibrational entropic term, you will get the delta S total. So this is not only just the entropy part, but it also has the temperature component also. So minus T delta S taken as a whole, you will see. So that is the value minus 17.8647 here. Okay. You can see the temperature here was set to 298.15 Kelvin. So very close to the 300 Kelvin temperature that is often used for calculations, right? So that is kind of what a binding free energy looks like. The different components look like in a calculation. Okay. So Professor Gumiha went over the folding patterns of different proteins, right? So certain proteins can fold from a, let's say, denatured state or an unfolded state to the native state in a very faster rate, and certain proteins can take a long time, so they can undergo a slow folding process also. So based on the prominence or the occurrence calculations for different residues, you can get to know that which type of residues are usually seen in slow folding versus fast folding proteins also. So this is the question that is exactly asking that you have this different combination of residues given, and which of these residues, the question is asking, will be seen predominantly in slow folding proteins. Okay, so not in fast folding proteins, but they will be seen in slow folding proteins. So can you um, get the answer? So I see answer C in the chat. So of course, these might have to do with the characteristics of the amino acid residues also. So the characteristic meaning that their hydrophobicity, right, or the polarity conversely. So that might play a part in these different proteins, right? The different folding patterns of these proteins. So basically, in this particular case, the answer would have been option A, alanine and leucine. Now, based on the information that is given, of course, you will see that alanine and leucine, they are characterized as hydrophobic residues, right? The other aspargine, glutamine, those are your hydro sort of philic residues, right? Polar residues, and then you have serine also. Lysine is a charged residue. Cysteine is your disulfide bond forming residue. But it turns out that if you do a comparison of the residue composition of slow versus fast folding proteins, you will see that residues such as alanine, cysteine, glycine, and leucine, um, for example, if you just take a look at the hydrophobic kind of residues, right? They are usually categorized there. So the alanine, glycine, and the leucine, let's say, right? So you will see that their composition is very, very high in slow folding proteins. Let's not take into account how this composition is calculated, but here you will see that 9.7 is value given for alanine in slow folding proteins, 9.5 for glycine in slow folding proteins, and then leucine is 9.2 for slow folding proteins. Conversely, you will see that their composition for the fast folding proteins is slightly lesser, right? 7.5, 8.4, 7.6, so on and so forth. Okay. Then if you look at again the other set of residues, sort of polar residues, you will see that their composition in slow folding proteins is slightly lower than their composition in corresponding fast folding proteins. Right? For example, your aspargine is 3.0 in slow folding proteins versus 5.8 in fast folding proteins. Okay. Similar case for your charged residues such as lysine, right? 5.2 versus 9.5 in slow versus fast. So it turns out that the fast folding proteins, they will be mostly prominent in your polar residues. Okay, polar residues such as your aspargine, lysine, glutamine, so on. And then slow folding proteins, they will have a higher composition, relatively higher composition of non-polar residues, such as your leucine, glycine, so on, right? Uh, and this happens because so the polar residues or the charged residues, let's say, the type of interactions that they form, it is usually uh, formed very fast, or they sort of form very short kind of range contact type of interactions. Okay. Uh, Versus if you look at the hydrophobic residues, so the hydrophobic residues you will see they very high prominently form long range contacts or long range sort of interactions. So we talked about the different long range contacts, long range orders in the earlier classes also. So basically, if you have the sequence, and then let's say one residue is situated at position 5. However, due to the complexity of protein folding, the residue 5 that is forming an interaction with, let's say, residue at the 48th position. Okay, so they are very far in the sequence. However, they are coming very close in the 3D folded structure. So that is a sort of long range contact formation. Okay. And they are very highly or more prominently formed by the hydrophobic residues. And because the protein folding, because these types of long range contacts takes kind of time to form, of course, the protein has to fold in a certain way for the residues to come together. So that is why these hydrophobic residues, they are sort of more prominently found in slow folding proteins. Whereas if you look about your hydrogen bonds or your charge-charge interactions, right, these types of interactions, so they are uh, very often times form in a nearby residue ranges. Okay, so residues that are very closely situated in the sequences, they kind of oftentimes form this type of interactions. Of course, you know that secondary structure formation needs hydrogen bonding, right? In that case, the residues are also quite in a close like manner, right, in a close position, situated in the sequence. So those types of cases, and that's why basically your polar interactions or polar residues, they are more prominently found in fast folding proteins, as far as the explanation goes. Okay. Uh, so the different proteins can fold in a slow or fast manner. Uh, and of course, if you go from an unfolded state to the native 3D conformation of a protein, what it's supposed to look like. So what do you think is the time frame required for this kind of folding, for a completely denatured or a completely unfolded protein to go to a completely folded state? What would be the time frame required for this type of change from a completely unfolded to completely folded structure? So it's a broad question. Milliseconds to minutes. So millisecond, you are kind of right about that. So the overall range of the time that will kind of look like this. Okay, it's microsecond to millisecond. It's not hours. Okay, it's only microsecond to millisecond time scale. Now, if you see the changes that occurs in the proteins, so for example, if I just take one example of the theoretical or computational part of that, which is biomolecular dynamics simulation, you can also investigate. Right? So usually what we do is that we integrate the position of the atom over certain time interval, right? And that time interval is usually 2 femtosecond taken due to the consideration of the bond vibrations and so on, the minimum time required for hydrogen bond vibration, those type of things, right? So we integrate after every 2 femtosecond the position of all of the atoms that constitute a protein. Okay. And then if you keep integrating, so if you take one snapshot and then calculate the next position and then next, you will combine all of that, you will get a trajectory. So usually when you combine all of this in combined all of this into the nanosecond sort of scale, right? So you have the behavior of the protein or any protein ligand complex in, let's say, nanosecond type of scale. It could be 100 nanosecond, 200, 500 nanosecond, up to, let's say, microsecond sort of time. So in this particular range, what you will be able to see is the protein ligand binding, of course. Okay, you can use this to calculate protein ligand binding. And towards the higher part of that, if you go further microseconds, you might also see the unbinding process of the ligand from the protein. Okay. Uh, and then if you want to investigate structural changes of the protein, so what happens is that you have to go from the nanosecond scale, you have to go upwards. Okay, you have to move towards certain microsecond sort of time frame if you want to see sort of decent level structural changes. Okay, so these could be, let's say, your loop conformational changes. This could be certain secondary structure formations. Um, in local on a local kind of level, right? This could be the difference in kind of domain structure in a way. So that will take your microsecond, the low microseconds, let's say up to, let's say, 10 microsecond, 1 to 10 microseconds. So you will see these sort of changes. But if you want to go from a completely unfolded to, again, your the native folded state, it will take again far more time than that. So it will basically go from the higher microseconds up to a certain millisecond scale if you want to see the entire process. So that is why it is very computationally exhaustive uh, kind of calculation to theoretically see what sort of changes um, are occurring in the protein. Okay, from protein folding to unfolding states, from an unfolded to the folded states. Okay, so it takes microsecond to millisecond time scale. So again, um, so the different uh, calculations or different analysis that you want to do based on your particular interest. So that's why you have to do different scale of calculations, and that was the point of it. Okay, I'll just one moment. Yeah. So what I wanted to show you, since we talked about the protein ligand complexes, complex formations, etc., right? Um, I will try to show you the conformations of protein ligand complex. I think we have also gone over that earlier, but this time we'll also introduce how to see, for example, different interactions that are occurring in a protein ligand complex. So I'll just explain this in terms of protein ligand complexes, but of course, uh, depending on the tool you have at your hand, you can do the calculations for protein ligands, protein nucleic acid, different sort of biomolecular complexes, right? Share the screen again. So hopefully you can see it. Um, okay. So remember earlier what we discussed about was one tool called UCSF Chimera. So UCSF Chimera is basically a visualization tool where you can investigate your complexes, protein ligand complexes, and do certain calculations. Okay. For example, here we have a protein. You will see the ribbon structure here. Let me just hide all of the other residues. We'll be able to visualize this in a better way. Okay, so here, if you see, if I highlight it by a color, you will see that the ligand molecule it is present inside of the binding pocket of the protein, right? So as you can see here, so it's selected. So that's why a green kind of outline is indicated here. But that is the binding pocket of the protein, let's say here. Okay. So inside of the protein, via molecular docking, we have obtained it. Now, if you are asked, okay, which residues are part of the interaction pattern or interaction sort of formation in the protein ligand complex in this particular one using chimera. So do you remember how we talked about this earlier? For example, let's say you have to just get a generalized idea about the binding region. Okay, so your protein 3D conformation is visible here, but which residues are forming the context, which residues are part of the binding pocket. So how do you characterize that? We have visualizer for the sequence also, but no indications are given here as to the residues that could be forming the context, right, in the protein ligand complex. So do you remember any trick to do that? So there are different selection methods in VCSF chimera that will allow you to select certain residues, certain atoms based on the different kinds of options that you give, right? And one of those options is the zone selection method. Okay, so the zone selection method, what it does is that uh, depending on what you want, so it can either um, let's say select the surrounding region of a particular selection of a collection of atoms um, that are coming within a certain range, let's say. Okay, so it can do either that, or it can select everything except the certain radius or certain range from your selected set of atoms. So that can be utilized here. For example, if we just go to the selection option, first we have to go to the structure and we'll just select the ligand. Okay, so the ligand was already selected, but you can select in this manner. You will see the yellow highlight here. Uh, if the ligand is here, so it makes sense that the surrounding regions of this ligand, okay, the amino acids that are present here, that will only constitute the um, binding site or the binding region of the protein, right? So what we can do here is that we can select a certain zone around this particular ligand, all of the atoms of the ligand, and then everything that is coming within this particular zone. So even if they have a couple of atoms of a certain residue that is coming within this zone, so you can say that, okay, this is forming the binding pocket, or this is coming within the binding pocket. And usually the cutoff, sometimes it is kept at 3 angstrom to see more closely kind of connected residues. Sometimes it is kept at 5 angstrom in a general sort of investigation. So what you can do is we can go to the select option, we can go to the zone option, and we can select less than 5 angstrom angstrom distance from the currently selected atoms. Okay, so if it's less than 5 angstrom and if we click OK, then you will see that certain other residues of this particular protein that also has been highlighted. Okay, if you go to the sequence viewer now, you will see that the green colors are highlighting certain residues, amino acid residues in the sequence. Okay, so these are the residues that are present within 5 angstrom range from the ligand molecule. Uh, and that makes sense that these residues will form the binding site of the protein. So if you have to give, let's say, general idea about the binding site region of a protein, okay, for example, in different docking tools, you usually have to specify which site you want to bind. And let's say you have a reference protein ligand complex that is deposited in the PDB structure, okay, that you are taking as control, something like that, okay. So what you can do is you can download that PDB, open it up using something like UCSF Chimera, and then you can select the ligand. You can select a zone of 5 angstrom around the ligand, and you can get the residues, and you can say the other tool that, okay, these are the residues that are forming the binding pocket in a general kind of way, right? And then the pocket will, the other tool will create a grid or something sort of that surrounding that particular region, and then you'll be able to do your docking calculations. Okay, but that is a general way of getting your binding sites. You can also visualize it using the actions atoms response. So I guess certain atoms are missing here in the structure, so that's the visualization. That's why the visualization is kind of getting in a fragmented way, but it's okay. Now, if you want to see what types of interactions are present in this protein ligand complex, okay, uh, then you have to go further than that, and you have to take the implementation of certain other tools. Okay, and one of those tools happen to be called leakplot. Now, this leakplot plus software, if you ask you to download the software, of course, you have to give your academic email address. You have to uh, probably register for it beforehand, and then you will get the password and the licenses from them also. Okay, and here it's basically a graphical user interface based tool where your protein and ligand conformations or your interactions will be shown in something like this, some sort of plot. Okay, so each of the interactions that are forming, for example, hydrogen bonds, hydrophobic interactions, they will be labeled in different ways. You will be able to see which residues are taking part in which type of interactions. And then it also has an associated tool which is called dim plot. So this is the tool to visualize protein interactions or domain interactions, interface interactions, so on and so forth. Okay, it also has something called antibody plot. I guess this is an extension of dim plot, as they say, for plotting antigen antibody interactions. But mainly what we will focus on here is the tool leak plot. Okay, so for leak plot, again, you have to download the software. Of course, I downloaded it. It was in a zip file. Extracting that, you will see that this leak plus sort of folder was created. Okay, inside this folder there are multiple different files. For example, one lib file, one .bat file, one .jar file. Inside that, again, different other tools and programs are there, but mainly we'll run the software via this .jar program. So if you go back here, uh, where the original confirmation or the original software is listed, right? So you can run this tool using a command like this. Okay, so you need Java for running it. If you do not have it, you have to install it. But this is the overall user interface for the leakplot plus version 2.3.1. So this is the latest one that they are providing. And here it's mentioned that to start you have to use the file, open and select PDB file, and there is also an instruction manual via this help option that is given here. But if you go to the file option, if you go to the open file option, if you go select the PDB file, you see that it has the browse mode here, or you can further enter a PDB code if you are trying to investigate something deposited in the PDB database. But here, let's say we have already done a molecular docking process using some tool, but we want to investigate the interactions. Okay, so you can go to the browse particular folder, and then here the complex that I showed you earlier in Chimera, so that is listed here as complex.pdb. And then if you open this up, you will see that we can run either leak plot or dim plot or antibody plot. Okay, now we are not interested right now in dim plot or antibody plot. Let's say we just want to see the leak plot. And here it has already detected that there is something called UNK. UNK is usually abbreviated form of unknown, and usually in a lot of protein ligand conformations or in a docking output file, if you save a PDB structure from there, the ligand is listed as UNK, three-letter code. Okay, so that means it is detecting our ligand. There are certain options here if you want to include residue ranges, include orders, filter orders, so on and so forth. But let's say we just want to run the default calculation. So you see on the background in the terminal, so it has checked the conformation, the structure of protein ligand. It has run different calculations, different residues, different interactions, so on and so forth. Right? It has analyzed the structure also, as you can see on the um, background. It has minimized certain statistics. There are different trials, different iterations of that, plotted H bonds, additional bonds, covalent bonds, hydrophobic contacts, so on and so forth. And then finally, what you have output is this one. Okay, so this is your complex.pdb where the UNK one is listed as the ligand. And then based on this, you can see the different interactions are forming between the different atoms and the different atoms of the protein or the amino acid residues also. Okay. Now for better visualization, of course, what you can do, since right now the fonts are not that visible, you can go to the edit option. There are different, uh, for example, options listed here. This on and off option, you can see the plot title, plot labels, etc. Whether you should those should be visible or not. For example, if I just untick this plot title and if I press OK, you will see that this complex that was written here earlier, that is gone now. Okay. So if you want to visualize that further, you have to go in this particular tab. But there are other options for size parameters also. Okay. Now, right now, let's say these are the hydrophobic residues. Okay, hydrophobic residues that are interacting with the protein. For example, this is arginine 54. If you can see it, if you want to increase the font size for that, then there are options here. Okay, so this is coming under the hydrophobic residue names umbrella, and right now the value is given as 0.500. So if you make it 0.800, what will happen is that it will increase the font size for all of the hydrophobic residues. Okay, now let's say the visualization is a little better. And one interesting thing is that you can also drag different residues from its position to different places. Okay, so it will not let you drag it to all the way, because that will not make sense where the interaction is forming. But if you want to do slow adjustment of the different labels at the different sort of representations, you can do that. Let's do some couple of size parameter changes. For example, the non-ligand residue names, let's also make it 0.8. Then the ligand residue name, okay, that is UNK1, so it's quite visible, I guess. Or if it's not, then let's make it 0.9. Then if you have the atom names, water names, water is not present here, so we'll not look into that. Let's say 0.6. Let's say 0.6. Let's say something like this. Okay. If we press OK, we'll see. Now it seems that the different atoms and their names are also visible in a better way. Okay. So if you do the adjustment, you will be able to visualize them better. So that is how you can investigate the interaction pattern of a protein and ligand using this today available tool called leak-plot. Okay. Now let's see if I ask from this particular interaction diagram, which residues are forming hydrogen bonds from the protein between the protein and ligand. Can you explain which residues, which amino acid residues, and then which atoms of the amino acid residues? To go here and look at the color option, right? You will see that the different labels have been given here. Particularly for bonds, if you look at the hydrogen bond, so the usual option or the usual default color for the hydrogen bond formation, that is using this green color. Okay. In this particular case, your arginine 155, that is forming a hydrogen bond, and then as well as lysine 164, so that is also forming a hydrogen bond here. Okay. Now arginine 155, you will see that the NH2 atom, that is kind of doing the bond formation, as well as the NH1 atom with the O2 and O3 atom of the ligand respectively. You have the distance also indicated here, 3.0 to 3.18. Okay. Here also NZ between the lysine 164 NZ and then the ligand O4. So in this ring kind of structure, so that is forming a hydrogen bond in this case also. And multiple hydrophobic interacting residues that are also listed in this particular case. Okay. So just a basic kind of visualization tool that is worth investigating, because it might be useful in different cases where you want to identify different protein ligand complexes and their basically interaction patterns. Okay, just wanted to quickly let you know that. So yeah. Yeah, so let's go back to the um, question and answer. So yeah, so this next question is asking that from an energy-based approach, which interactions can identify the binding residues in a protein ligand complex? So remember that when we talked about the overall binding free energy or the binding energetics, so certain types of interactions that constituted the overall binding energy that was listed. Okay, so do you remember which type of interactions were there for energy calculations? Particularly if you see, let's say outside of the solution energy, so there was a part of polar and non-polar solution free energy. But outside of that, there were two energy terms that was listed in the overall MMPBSA calculation in a table that I showed you earlier. Okay. So do you remember which interactions were there in that case? So one particular interaction was calculated using something called Lennard-Jones potential, if you remember, and then the other one is uh, calculated using the Coulomb's law. Okay, Coulomb's formula. So of course, these two interactions are van der Waals and electrostatic interactions, quite crucial in calculating the binding free energy and to get the overall sort of interaction pattern and energetics of the complex. Okay. Um, Now, of course, your binding site that are composed of different residues. Okay, but it is possible that for different proteins, certain amino acids, their composition could be higher on the binding interface or the binding regions. Okay, so depending on your what this particular protein wants to bind to. Okay, for example, let's say if it has to bind to, let's say, a negative kind of charged molecule like your nucleic acids. Okay, so then that means that on the interface, the prominence of charged residue or the composition of charged residue on the interface, that will be higher. So that's one thing, right? So if your ligand uh, that is sort of having a very high like polar sort of environment, so then it will be buried in a hydrophobic group or polar group. Okay. Now based on that, you can devise different questions. Now here it is asking that uh, you have a protein and you have been given that a certain amino acid residue, for example arginine, the number of it in the overall protein is 2414, and it seems that 366 out of those 2414 arginines, they are involved in binding and they are present at the interface um, of this particular protein. So what is the binding propensity of arginine here? So again, a very simple calculation. You have to just calculate the propensity, and remember that we used to calculate propensity and these sort of things using fractions, right? And you can convert those to percentage also. This is 0.15. So for calculating propensity, of course, you need the number of arginine that is present at the binding site. You need the number of total arginines, of course. And then if you do 366 by 2414, you will get 0.1516, right? That is the binding propensity. In terms of percentages, it will be 15.16%. These sort of simple calculations can come your way. Just be aware of those, right? Now here one interesting question is there. So it is asking that computer-aided drug design cannot work on which of the following sort of scenarios. So it is asking about the known or availability of the protein and ligand structures or conformations. Okay. Of course, it's kind of straightforward here, but which option should the correct answer be? So if you do not have a protein structure known, there are certain steps that we also discussed earlier which can get you the protein structure. Okay, so if you have the sequence of the protein, um, you can go for different tools, for example AlphaFold, or you can go for a homology modeling approach where you will be able to get the protein structure, and then further you can try to optimize the structure, try to minimize the structure overall to make it to a decent level where you can calculate a molecular docking approach, which is a part of computer-aided drug design, right? So even if you do not have the protein structure, you can get it using different tools. For ligand structure, of course, if you do not know that as well, so that will not work in this case, right? So again, if you do not have the structure, you have to sort of draw the structure using tools like ChemDraw, and then from the output file or the output structural file, you have to again further perform minimization calculations to kind of have the structure at a certain energy minimized level of the topology of the structure, for the topology of the structure, so that you can do further calculations. But in this particular case, the better option or the most correct one will be the unknown ligand and unknown protein structure. If you do not have any of those, you can not perform a computer-aided drug design calculation, basically, right? Now, there is one way where this is possible, um, is that if you use, let's say, machine learning algorithm where you have already trained, not based on any physics-based, docking processes and such things, but let's say you have already available case, or we have already available information regarding certain IC50 values or KD values of different ligands against the same target protein. So if you can train your model using that, and then if you have, let's say, the smiles of the ligands also, then perhaps you can convert those smiles into features, and you can train the model using that. And now your unknown ligand, if you do not have the 3D conformation, but you have the smiles of the ligand, so again you can extract features from that, and you can be able to predict what your interaction score or what your binding affinity would be based on your already trained model of machine learning. Okay, so that is one possible way that you can go forward in that case, but it's better that you build the structure of the protein, and you can also do the energy minimization, etc., for both protein ligand, uh, and that would give you a better option, basically. Okay, now in drug discovery, of course, we often talk about the admit properties, right? So what does admit means? Can anybody explain? And why is it necessary part? And how can we calculate it? It's perhaps.
Sir, can you repeat the question?
So in drug discovery, for example, there is one term called calculating the admit parameter or predicting the admit parameters. Okay, ADMET, right? So what is admit, ADMET? And then does any tool come into mind where we can predict such parameters, basically? So ADMET is basically your absorption, distribution, metabolism, excretion, and toxicity. Okay, so these are how your drug would behave in a physiological environment. And of course, there are different parameters that you can calculate under each sub umbrella. And overall, taking all of this into account, we have to see whether your drug is suitable to use for treatment, right? Because it could be that the drug is showing different toxicity. It could be that it does not have that much bioavailability. It could be that it lacks the uh, intestinal absorption that is required, right? It could have off target effects if it is able to penetrate, let's say, blood-brain barrier. That could also pose different problems. So there are different parameters under the overall ADMET umbrella, and then you have to calculate all of those to get a good idea about if your drug is a suitable candidate for therapeutics or not. Now, under those umbrellas, like the overall calculation of ADMET, oftentimes we see we basically take into account certain established rules, okay, for selecting suitable drug candidates. One of those rules, it is the Lipinski's rule of five, which has been since a very long time, and it basically describes certain parameters and what the ideal value ranges of those parameters be. Basically, a cutoff where if the particular drug molecule is going over that cutoff, then it's possible that that will not have the suitable properties for the drug candidate. Okay, so in Lipinski's rule of five, there are certain those kinds of parameters that he talked about, which is oftentimes taken into consideration. So, does anybody know what the different uh, the criteria for the ligand molecules are in the Lipinski's rule of five? So, Lipinski's rule of five talks about the molecular weight. It talks about the number of hydrogen bond donors and the number of hydrogen bond acceptors in the ligand, and also it talks about this log p value. Okay, so what is this log p? It's the octanol water partition coefficient. Okay, now what is the maximum value of log p that the question is asking in Lipinski's rule of five? You will see that the maximum value of log p that is allowed in Lipinski's rule of five is 5 cost. Okay, so it's called Lipinski's rule of five because all of the sort of cutoff values that is given here, they are multiples of 5. So that's why it's called Lipinski's rule of 5. There are only 4 criteria, but since all of them are multiples of 5, the cutoff value, so that's why it's given as Lipinski's rule of 5. So it's saying here that the molecular weight should be less than or equal to 500 Dalton. The log P or the octanol water partition coefficient, the log value of that should be less than or equals to 5. The number of hydrogen bond acceptors in the ligand that should be less than or equals to 10, and the number of hydrogen bond donors in the ligand that should be less than or equals to 5. Okay, now here it is saying this is taken from a particular tool, online tool. I will talk about it also. So here it is saying that how do you interpret the results. So if two properties are out of range, let's say it has 13 hydrogen bond acceptors, 7 hydrogen bond donors, right? uh, then it is possible that there would be a poor absorption or permeability for this particular ligand. Okay, now it's saying that one violation is acceptable in this particular case. Let's say your molecular weight is not within 500. Let's say it's going to like 600, right? 600 Dalton. But the other parameters are within range. So then one violation that is quite acceptable, whether your drug should be suitable or not. Okay. So empirical decision is two violations. Um, less than 2 violations, so that is basically green. A green signal. If it is greater than equals to 2, then that is poor signal, basically red signal. So what is the N-octanol water partition coefficient? So it is also called KOW. So it denotes the partitioning of substances between N-octanol and water. Okay. It is also denoted as P, but it's basically an indicator of the relationship between a particular compound's lipophilicity, which is fat solubility, and hydrophilicity, which is water solubility. And if it is, for example, exceeding 1, a value exceeding 1 KOW, that is suggesting higher solubility in fat-like solvents, for example, in octanol, whether a value, while a value below 1, so that will indicate a greater solubility in water. And here, the cutoff for log P value for it to be suitable drug candidate is given as 5. Okay. So I believe in database when we talked about not database, the different web servers, I also mentioned this particular one. So this is called Admit Lab 3.0. Okay. So Admit Lab 3.0 is one of the tools, online available tools, where you will be able to predict beforehand the admit properties of certain compounds. Okay, you can do, let's say, single by single calculations, or you can do bulk calculations by providing all of the ligand smiles together. Okay, so here under the services, of course, you have admit evaluation and admit screening. If you go for admit screening, of course, it will ask you to load a molecule file or a list of smiles you can give here. Okay, so maximum 1000 simultaneous calculations it allows. but we'll just do a simple admit evaluation. Um, and then here it is asking for a smiles. Now, how do you get the smiles here? Well, if you know the compound, let's say if it is listed in PubChem, uh, then what you will see that they also list the smiles of those molecules in PubChem also. Let's say we have one particular molecule here, fostamatinib. So it's listed in PubChem, of course. It's a drug which is FDA approved for immune thrombocytopenia, so in autoimmune disorders. Okay. If you go down here, of course, a lot of information are given here. You can download the 3D structure, 2D structure. 3D conformer is basically used for molecular docking and these sort of calculations. So you can get this data from PubChem also. You can get the molecular formula, the different synonyms, so on and so forth, and other information related to it also. There are also different tabs for toxicity. For example, hepatotoxicity and effects during pregnancy, lactation. These sort of different parameters, safety and hazard parameters, pharmacology, etc. They are also listed. You can take a look at it. But let's say if you have the smiles, if you want the smiles and you want to calculate it using ADMET so you go down here, and here is the particular parameter that you want. So this is the smile notation for the ligand. Okay. Uh, so the smiles notation for the ligand, you can copy it from here. Let's say for just example, you can paste it here and you can submit it. And usually single calculations are run instantly. Does not take that much time. And what it gives is it gives the list of different physicochemical and pharmacokinetic properties for the ligand molecule. Okay. Firstly, if it is listing your overall, for example, molecular weight, volume, density, hydrogen bond acceptors, donors, etc., the different number of rotatable bonds, number of rings, flexibility, stereochemical properties, and then you have your log P, log S, pK, pKa for acid and base, etc., so on and so forth. So different parameters are listed. But alongside that, you will see that different uh, metrics related to absorption, distribution, metabolism, medicinal chemistry also, excretion and toxicological properties. So that is listed in this particular case, right? And based on all of this, you have to not only select whether this molecule is suitable for further use, but you have to also select the mode of administration, how you will go forward with the therapeutics of this particular molecule using these sort of different calculations. So usually these prediction models are used on the earlier part of a computer-aided drug design pipeline. So that means that you validate it using this sort of predictions, and then you go for your computational calculations, and then when you do experimental calculations after that, if you do your in vitro in vivo experiments on those stages also, you have to sort of calculate these sort of parameters, toxicity parameters, absorption, distribution, etc. Because ultimately the experimentally validated ones you will be requiring for your final approach of using this drug, right? Now, interestingly, here you will see uh, that this drug quite violates certain parameters. For example, it has respiratory toxicity, certain type of hepatotoxicity, drug-induced nephrotoxicity, etc. Okay, so it seems that even though the molecule is FDA approved, it still shows certain red sort of indicators or red signals in different toxicity parameters also, and as well as in different distribution parameters, so on and so forth. So that means that if you have a particular drug candidate or a drug molecule already approved, it's not that it's the best one, okay, and that no drugs can come after that. Still, the limiting factors of the drugs, the side, off-target effects, let's say, or the side effects or the adverse effects such as toxicity or poor distribution, so on and so forth. So these parameters can still be accounted for uh, in designing, let's say, potentially more suitable drug, a potentially better sort of drug candidate. Okay, that's why if a drug is already there, it's not the end of all. You can still try to find a better one in this case. And interestingly, it seems that in the physicochemical property calculation, the molecular weight of this particular molecule, fostamatinib, that is 580.15 Dalton. So that's one violation of Lipinski's rule. Okay, so it's going above 500 Dalton as mentioned in the rule. And you will see, if you go to this particular chart, you will see that the hydrogen bond acceptor was listed as less than 10 and donor was listed as less than 5, less than equals to 5. Here it seems that the number of hydrogen bond acceptors in this calculation is 15. Okay, so that means it's going above 10. So now there is two violations of Lipinski's rule in this drug. The hydrogen bond donor is of course 4, and then your log p value is 1.6, which is less than 5. But still two violation, as we looked at earlier, this particular tool suggests that less than 2, only one violation, it is acceptable. So it turns out that in the recent years, so different researchers, they have analyzed the set of approved drugs and the state of different drugs at certain stages of clinical trials and what effects they have after the basically the usage and distribution of those drugs, right? And they have also correlated those with the different physicochemical properties and parameters of the compounds. And what they're saying now is that since Lipinski's rule is a quite old rule, so it's already established a long time ago. At the time, it was basically seen as a kind of standard benchmark for selecting your drug candidates. However, now based on the different selection or already selected drugs and the approved drugs and kind of drugs that have shown good results, so they are saying that Lipinski's rule is not the end of all, basically. So compounds can still violate um, Lipinski's rule, and they can still give you good results uh, despite violating certain terms. And you will see that the different companies that make the drug molecules, for example, the pharma companies, right? They have oftentimes their own set of calculations. They run their own set of metric analysis that they believe is a more suitable one for characterizing the drug candidates and selecting whether this is a good one or not. For example, your Pfizer's, they have your Pfizer rule, GSK rule. So these different types of rules are present. I believe it is also listed in the medicinal chemistry tab for the Admetla 3.0. So you will see quite well that Lipinski's rule is being rejected here. However, it seems that Pfizer rule is accepted here. So Pfizer is a big company, right? And if you go to this information sort of option, you will see that compounds with a high log P and then low TPSA value, they are likely to be toxic. So it's not considering any other factors, but it's considering the log P and the TPCA value, the topological polar surface area value. So these different companies, they have devised their own rules based on their understanding. And basically that's all. So it's not easy to select a drug candidate from a potential list of candidates, but you have to take into account uh, the different parameters that are coming from the calculations. Okay. So that was just one tool that I quickly wanted to go over again. I guess we will go forward with the other ones. Okay. Yeah. Okay. So now, as far as the protein folding concept, so Professor Gomiha went over something called chevron plot. Okay. So here the question is asking what is chevron plot used to investigate. So any what could be the good answer? What would be a good answer for this question? Chevron plot is used to investigate what? So chevron plot is particularly used to investigate thermodynamics of the protein. So it's more properly the kinetics of protein folding. Okay. So kinetics of protein folding, that is investigated using the chevron plot. Now you see in this particular figure on the right hand side, right? So it is an experimental protein folding landscape, sort of in a two-state uh, protein with no intermediate. So what are those two states, the two main states? You see that one state is sort of termed as D here. Okay. Now this D stands for a denatured protein. That means it's completely unfolded. And then on the other hand, you see one state is termed as N here. So this N stands for a native state of the protein, the native folded state of the protein. And of course, certain delta G value uh, gives the energy difference between D and N, the denatured and the native protein. And it seems that the energy level of the native protein that is at a lower energy value than the denatured protein, right? So that means that upon folding, certain energy is liberated from the system, so that the system is becoming more energetically minimized and more stable. Okay. Now, when the protein goes from the D to N end state, it's not that it's just directly energetically going from here to here. Does not work in that way, but it goes via multiple intermediates. It could be, or it could be something called the TS, which are the transition states. Okay. So here, for example, in the folding process, you see that denatured protein goes towards the transition state, and then after that, it comes back to the native state, let's say. So what are these transition states? In this particular figure, it says that for a two-state protein with no intermediates. Okay. So are transition states not intermediates? How do you characterize the intermediate or a transition state? Are they different or are they same?
Sir, actually, they are different because transition state is a fleeting moment. It cannot be calculated, but intermediate is something that you can calculate or you can see along the reaction side. So just for example, I'm not sure about the chevron plot, but I'm quite understand about the basic kinetics of protein and the transition state of. From product to substrate is the point where the product is converting into substrate.
Yes, yes, exactly. So the transition states, as you say, they are quite fleeting moments. And on plot one, you will see this is sort of, as you said, a product to substrate, a starting material, a substrate to product formation based on the reaction coordinate and the energy value here. You will see that the starting material, of course, it goes to the product, but it goes there via two different transition states, TS1 and TS2, as indicated here. So the transition states are basically the states where a lot of bond formations and bond breakings are happening. Okay, so that's why they are very high energy and very fleeting states. You are not able to capture those states in the standard sense. Okay, but the protein has to go through these states to ultimately reach where it needs to be, basically. You will see from the starting material. So this EA is the kind of the activation energy that goes to the transition state 1. And then after that, between transition state 1 and transition state 2, you will see there is a dip in the energy value. A valley is seen here, which is termed as I, or intermediate. Now, these intermediate steps are where your conformation or your particular, whatever you are kind of analyzing here, so that can be captured. Okay, so that can be captured, and that tells you a particular path via which your protein, if you talk about it in terms of protein, your protein is going from a folded to unfolded, or an unfolded to a folded state, let's say. In the intermediate state, that sort of characterizes a metastable conformation, which is possible, which is coming somewhere in between the initial and the final product. And you can able to capture it, you can able to analyze it, you can see what is going on, what kind of changes are there, so on and so forth. The transition states, they are very high energy, uh, very fleeting, as you said, states, which you are unable to capture in the standard sense, right? Now, some links are also given here. But if we look at the chevron plot, as Professor Gomiha mentioned, so chevron plot is basically a folding sort of kinetics plot. Okay. Now here you will see that this plot has different black dots, of course, and each black dot, as mentioned here, that represents a separate kinetic experiment. Okay. Uh, Now, what are the separate kinetic experiments? So they will be coming under the umbrella of folding experiments and unfolding experiments also. Okay. Now, on this particular axis, the x-axis, you will see the concentration of certain compound is given. Okay. So in this particular case, the concentration is given in molar for guanidinium chloride. So guanidinium chloride, that is a very strong denaturant, and if you start adding that to a protein, of course, it will um, go towards unfolding. Okay. And what will happen here, if you progressively add, let's say, different concentrations of guanidinium chloride one after another, then you will see the different unfolded states where progressively more kind of unfolded states will get a trajectory of that from this kind of unfolding experiments. Conversely, if you have already unfolded state, and then you start to dilute the sort of the solution or the medium that is in there, then the guanidinium chloride's concentration will progressively reduce, right? In this particular case, if you just keep it diluting it, and then the protein will slowly sort of fold towards the native-like structure back. Okay, so that will be your folding experiment, let's say. And on the y-axis, you have your certain parameters regarding folding or unfolding. It's k, ln k value. And then here, as it is seen, what I just told you. So unfolding experiments indicated by the red dashed line. So this is the red dashed line that is talked about here. They involve mixing folded protein with denaturant solution, guanidinium chloride, to monitor the changes using structural probes like circular dichroism or intrinsic fluorescence. Okay. Now in the folding experiment, of course, which is the blue line. So this blue line is a folding experiment. Uh, it is where a unfolded protein in high denaturant solution that is mixed with buffer. So basically you are keeping diluting it to observe the folding using similar sort of optical probes. Now, of course, it is mentioned that folding is slower in the presence of denaturant and unfolding is faster with higher denaturant concentrations. And the logarithm of the folding and unfolding rate constant, which is ln kf and ln ku, so that is directly proportional to the denaturant concentration. And then you can also extrapolate this constant to zero denaturant concentration, provides that will provide your kf and ku in water that will be used to determine the stability of the protein. So basically, if your dotted lines are indicative of the experiments, and let's say you just extrapolate this data and you see where it matches on the folding parameter, then you will be able to find out what was the particular parameter at zero guanidinium chloride concentration, right? Similarly, for this particular experiment, the blue one, you can again get the same parameter for ln kf of H2O here, what is mentioned. Okay. So from that, what you can do is you can calculate a free energy. So del G equals to RT of ln kf divided by Ku. And that will be able to see the stability of the protein in a way. Now, the chevron plot that also has the other characteristics. For example, here, this is the red one is for an isolated domain, and the black one is for a multi-domain construct. Okay, it seems that the domain, if it is present on a single sort of state versus a multi-domain state, so the domain fold at the same rate, but if it comes down to the multi-domain state, the unfolding of the protein is more slow in the case of a multi-domain sort of conformation versus a single domain conformation, which is given as these differences between this red and then the black line here. Okay. And then similarly, we'll also see that a domain unfolds at the same rate in a multi-domain context, but folds more quickly. So these are again different experiments based on which we can determine the domain-domain interaction effect on the folding of a protein. And then it is also saying that when the neighboring domain offers favorable interactions to both native state and transition state, what will happen there? It will happen is that if the neighboring domain stabilize the protein by both speeding folding and slowing the unfolding. So you can take a look at that via this link given here, and you will be able to better understand the chevron plot. Okay, but now we come to the other parameters which also kind of has a correlation, let's say you can say, with folding. And one of these parameters is contact order. Okay, now it is saying that particular protein has a contact order of 8.7 and the other protein that has a contact order of 5.7. So which one will be folding fast? Okay, so which one should be folding fast here? That you can calculate based on the contact order calculation. And here in this case, the fast folding protein that will have a low contact order of 5.7. Now, if you remember on the other questions, we also saw that the hydrophobic sort of amino acids, which predominantly form long range orders or long range contacts, right? They have a tendency to fold slowly. Okay, because those type of contacts in the native state, that takes a long time to form. Similarly, if you have, let's say, majorly charge-charge interactions or nearby hydrogen bond formation, so on and so forth, if you have a prominence of those amino acids, they will relatively fold faster. Now, the contact order is kind of sort of similar parameter which tells you about the formation of contacting residues depending on the sequence separation between the residues. Here it is saying that the sequence separation between contacting residues. So the contacting residues meaning that in the 3D space, the distance between those residues are coming within a certain angstrom value. And that angstrom value is here, for example, the one that Professor Komiha mentioned is 6 angstrom. Okay. So if residue I, residue J, they are coming within 6 angstrom distance, that means they are contacting, they are in contact basically. And the sequence separation between those is taken into account here. And what it says is the contact order will be summation of the delta Sij, which is sequence separation, divided by the total number of residues and the total number of contacts that have been formed in this protein. And what happens is that if there are more long range interactions, okay, so if there are more long range interactions forming in this protein, so that will give it a high contact order value. And if the high contact order value is there, its folding is seen to be slow if you correlate it with folding rate. Okay, and if the protein are predominantly short range interaction forming, then you will see that the contact order is lower, it's lesser, and it is folding at a faster rate. So basically, this is a relation between the long range interactions, contact order, and the protein folding also. So that's what this question was asking. And since protein B had a contact order of 5.7, that means it would be the faster folding one. Okay. So based on the different contacts forming, of course, you see that you can also correlate them with the protein folding pattern or folding rates also. Yeah, please.
Sir, the distance separation 12 Armstrong, something like that.
Mhm. Uh,
Wait, just a second.
this one.
Huh.
Yeah.
So distance separation,
Mhm.
what 20 Armstrong was from C alpha, right? I got a call and I got disconnected.
Uh, uh,
So my question is that
yeah.
there was Professor Gromeha
Uh-huh.
talked about some distance separation regarding 12 Armstrong. I
So I have
it
to. Okay. 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 So I have to see
so
that in
I
context what he talked about. But in this particular
okay.
case, for contact order calculation, so the threshold for finding out whether two residues are in contact with one another. So that is 6 angstrom given here. Okay. 12 angstrom I think would be a
Okay, so
too larger distance for that. So
from
I think, yeah, yeah.
from C alpha something like that. It was. I'm not sure about my question also, but there was something that was 12 Armstrong.
Okay, okay. So I have to
I may be wrong. I don't know.
check it. So 12 angstrom is quite large distance. If you see C alpha distance also. Um, so again, then I don't think 12 angstrom should be one. So I have to again check what Professor Komiha exactly showed there.
Okay, so
But yeah,
okay, so sir, I don't worry about it. I think so. I might be wrong. I might have
no, no,
uh
it's okay.
seen it somewhere else. Sorry, sir.
No, it's fine, it's fine. So I think it could be in a different context also, or it could be in the same context. I will have to again check. But what in this particular case, as per the contact order calculation, the contacting residues, let's say they have a 6 angstrom distance. Now it's quite possible that let's say different tools have been devised to calculate the contact orders. It's quite possible that they take the angstrom value at a different sort of range. For example, they could have 5 angstrom distance as the threshold cutoff, right? They could have 4 angstrom. They could have no, 4 is too low, I would say. But let's say 5, 6, or 6.5. These kind of values they can take. But here, based on the general one, I believe it's kind of categorized in 6 angstrom cutoff value. Okay, but that's just the base concept here. So nothing to worry about that. Okay. Okay. Of course, you saw that the contact order that was correlated with the protein folding rate. Now, what will be the long range order? How will that be correlated with the protein folding rate? It's asking. So positively or negatively? We just saw that proteins that are more prone to forming long range contacts, they will be slow folder proteins, right? So that means since long range contact was connected to long range order, of course, its calculation are involved. So they will have negative correlation between between them, the protein folding rate and the in the long range order. And this was the kind of formula for calculating long range order. This is a summation of Nij divided by capital N. So Nij is basically it could be either 1 or 0. If the sequence separation between uh, the two residues that are forming contact, that is greater than 12. So the sequence separation here, if that is greater than 12, so that means they will form a long range contact. So that is given as 1. Otherwise, it's given as 0. So it's not exactly um, the distance, the 3D space, but it's the sequence separation. And for example, here in this case, as Professor Pumiha talked about in his slides, so here the C alpha atom distance is the cutoff is taken as 8 angstrom. Okay, so as compared to the previous 6 angstrom calculation. So there are different discriminations between different approaches or between different tools. Um, for example, in long range order versus the previous calculation for contact order. But that's just how it is. Okay, so you have to take default values and you have to choose between what the current research scenario is kind of exploding, what is the best one to take as. Okay, so that was one calculation. And then as we saw that the contact order and the long range order both can give correlation with the protein folding rate. So now it stands to kind of question that which of them could be better correlated with the protein folding rate. Okay, contact order or long range order. Now to investigate that, it is found out that the long range order seems to be better correlated with the protein folding rate as compared to the contact order. So here, in terms of different secondary structure characterized proteins, for example, all alpha, all beta, or mixed proteins, you see that the contact order and the long range order, they have both been evaluated against the protein folding rate, and the correlation has been calculated. And you see that in case of all alpha protein also, the contact order gives upwards negative correlation of minus 0.56. However, the long range order in this case, it is minus 0.72. Okay. For all beta again, minus 0.46 for contact order. For long range order, it is minus 0.92. And again, for mixed type of proteins, so it could be alpha plus beta or alpha by beta. So R is minus 0.82 for contact order and minus 0.86 for long range order. In this case, there is not that much difference. The difference is not that big, but where it is primarily dominant in one type of secondary structure, you will see a markedly larger correlation if you calculate via long range order. Okay, it's always negatively correlated, but the correlation extent is better calculated using the long range order. Okay, so that's one question there. And of course, since contact order and long range order, you saw that both of them are similarly correlated with the protein folding rate, right? So it stands that there would be relationship between those as well. And of course, they have a positive sort of correlation between those, because that makes sense, right? They among those, they are positively correlated, and then both of them are negatively correlated with the protein folding rate. So it quite makes sense. Now, here the question is particularly asking, uh, we talked about the different classes of the protein, right, based on the dominance in secondary structure. For example, all alpha classes will have mostly alpha, all beta will have mostly beta. So which will be the slow protein folding, or slow protein folding will be observed in which type of classes of proteins, right? So if you remember, the alpha helices, uh, the pattern of hydrogen bond forming there. So there you will see that uh, I with I plus 4 residue that is forming a hydrogen bond, right? In the case of beta sheets or beta strands, you will see two adjacent strands, they are forming hydrogen bonds with one another, right? Now in alpha helix, you will see that very close by hydrogen bond formation is there for the helix to form. Whether in beta sheets, distant residues are forming hydrogen bond in this case, right? So based on that, you will see that since this sort of secondary structure formation in the all alpha all beta class, where it will be mostly dominant in beta sheets. So for that, for the folding to occur, of course, distant residues after forming beta strands again in a pattern of beta strands, they have to come together, they have to come closely, right? But since these are much distance, like high distance interactions that are occurring in the all beta class that are primarily dominant there, so they will take larger time to get into that conformation basically. So that means that for those types of proteins, for the case of all beta class of proteins, you will see a more slower rate of protein folding because distant residues, they need to come closer together to form the type of hydrogen bonding pattern that is mostly prevalent in this type of class. Conversely, for the all alpha class, you will see that they will have a relatively faster rate of protein folding because again the hydrogen bonding forming residues are much nearby as compared to the beta sheet or beta strand dominant. Okay, just to quickly recapture, these are the all alpha proteins dominant in alpha helices. These are the all beta proteins mostly dominant in beta sheets. And based on the segregation, we saw alpha by beta or alpha plus beta type proteins are also possible. Okay. And then Professor Gomiha also mentioned one database where it has the binding affinity for protein-protein complexes after mutation. That information is given. So which database is that? So it's a database that is in IIT Madras server, right? Developed by Professor Gomiha in his lab. So it's called the ProxyMate database, right? So you can search for this database online. You will get the information. They have different types of data available, different types of binding affinity, mutational data, etc. So you can look it up. It's available in the IIT Madras server also. Okay. Yeah. So I think that is all that I will talk about today. Sorry that I had to prepone this class, which was supposed to be on Monday. And then for the next Monday's class, so the Monday that is coming after this one, so that one will also be postponed to some other date, which I have also communicated to the NPTEL platform. So again, we'll see, we'll look forward to discuss in that class also. And then of course, the other TA session should be scheduled as per the correct, as far as the regular schedule. So previous question regarding protein folding, which one? The all alpha, all beta, this one, I guess. Okay, so here basically what happens is that, let's say you have a protein that is, okay, let's just go forward one slide. Let's say you have a protein that is very highly dominant in alpha helices, right? Now in alpha helices, we know that I and I plus 4, this type of hydrogen bonding pattern is seen. For example, one, if residue 1 is here, then this residue is probably forming hydrogen bond here, here. So this type of short range hydrogen bonding pattern is seen in all alpha type of proteins, right? Because they're dominant in alpha helices. So this is the measure of the interaction that will be observed here, of course. Now, the all beta proteins, if you look on the other hand, so the different beta sheets, or sorry, the different beta strands, they need to basically come in a very close proximity to form the hydrogen bonds, right? Uh, so, like let's say in this kind of pattern, right? Now, here what you will notice is that since let's say this a particular residue that is situated here that is forming a hydrogen bond with this residue, so there is a larger sequence separation, um, the amino acid sequence separation between these residues. which will be present in a beta sheet. Because of course, if you look at it, so if you look at, let's say, long kind of beta strand, let's say, so here, for example, the residue that will be situated here, that needs to form a hydrogen bond with the residue that is situated here, right? And there is a much gap in the sequence in this particular interaction forming residues, right? So that means what is happening is that for the particular type of interactions to form, the hydrogen bonding patterns to form in an all alpha protein, it's quite uh, kind of easy, you say. It's a faster process because the residues are already close by, which needs to form this sort of interaction patterns. So that is why the folding of these sort of proteins for alpha proteins, that is faster, because the residues that will form the interactions, the primary hydrogen bonding interactions, that are very close by. However, on the case of all beta proteins, you will see that the residues, they are not close by. Not all of the residues that will form the hydrogen bonding contacts here, they are close, right? They are separated in a large sequence gap, let's say. So if you have an unfolded protein, let's say this is your unfolded peptide or whatever, and if this residue and this residue is supposed to form an interaction, and let's see if you have other ones, which is mostly dominant in beta sheet, while let's say this residue and this residue is supposed to form an interaction. So of course, it makes sense that the alpha helix dominant proteins, where the interacting residues that are supposed to interact that are nearby, that will form faster, because interactions are forming faster, they are close by, right? Whereas in this case, where it is mostly beta sheet dominant, so these residues need to first come closer before they can forming the contact, right? Before they can forming the hydrogen bond for this type of proteins. So then you can correlate that, of course, with the folding rate, and it makes sense that the all alpha proteins will be faster folded than the all beta proteins, because in the all beta proteins, distant residues need to come closer to form the type of hydrogen bonding pattern that the beta sheets have. So that's all. Just basically the distant residues, if those distant means if high distance interactions are present in certain proteins, so they will be slow folder. If majority low distance interactions are present, let's say in a folding for different secondary structures, so that will be faster folding proteins. So that's just the logic of it, basically. Okay, yeah. So I guess I will wrap up here. Again, as always, thank you all for joining. Go over Professor
Thank you,
Bhumiha's.
sir.
Yeah. Please go over Professor Bhumiha's lectures also, and then attend the other TA session that will go on, I guess, regularly on Tuesdays. Yeah, and then if there is any change um, in the TA session for myself, I'll just inform the NPTEL team, and they will further communicate with you. Yeah, so thank you everyone, and we'll meet in the next class. Okay, thank you.